For each programming assignment, I will give you some background information and I will describe the types of functions that you would need to solve the problem. For this week's assignment, I'm going to review the programming concepts that you would need to solve these types of problems, okay? All right, for the programming assignments, you are welcome to use other programming languages. I will base my explanations for MATLAB. If you want to use another programming language, you would have to sort of transfer what I'm describing to your own programming language. Okay. You may want to use Python, for instance. I'm not providing any Python code. It's going to be your job to translate my MATLAB code that I provide to Python. Okay. How many of you do want to use another programming language? None? All right. So you're welcome to use Python if you'd like. At the end of your assignment, my main requirement is that you produce a PDF document demonstrating that your program works. Okay. We will have at least one assignment where I will require everybody to use Python. Okay. I will give some background so that you can start using Python, and we will look at the BioPython library, which provides methods specific to bioinformatics. Okay. In this first assignment, you are asked to take a DNA sequence and find out the gene included within that DNA sequence. So if we are given a sequence like this, that's a DNA sequence. Parts of it is encoding a gene. The other parts are not. So we would like to find out which portion of this sequence encodes a gene, where a protein is being produced from it. When you review your molecular biology basics, you should already know that we have DNA that is being transcribed into RNA, and then RNA is being translated to proteins. You are given the DNA, you are asked to find at the end of this assignment the protein that's being encoded by the DNA. The entire DNA segment is not translated to a protein, only a portion of it. The enzymes that are involved in this process recognize a certain signal within the sequence, and they start translating into a protein from that signal. Okay. And the simplified rules that we're going to have for the assignment are the following. We are going to define a gene as a reading frame, which starts with an AUG codon. A codon is triplets of nucleotides, so 3 characters. And it may end with one of the stop codons. We have 3 types of stop codons. So each of these signal the end of the translation. We can have a UAA, UAG, or UGA. So if we have a signal, a long sequence, and we encounter an AUG here, and a UAA here, and say a UGA here, translation will start from here, and it will stop when it finds the first stop codon. Okay? All right. Before we find and translate the longest open reading frame, I have divided the problem into a smaller task, into a pre-processing step, where you would take a DNA sequence, the coding sequence, and identify the non-coding sequence from it, the messenger RNA from it, and the protein from it. Okay? So let's do this by hand first before we start programming it. DNA is a double helix. composed of nucleotides. The segment that is given to your function as input is the coding strand. So one of these strands is composed of A, C, G, T, G, A, and finally we have A, T at the bottom, this end. Okay, so that's our coding strand. Oh, I spiral to the other side. So this one is non-coding. This one is coding. All right. In the non-coding strand, the sequence is going to be complementary. Complementary means you look at the character. If it's an A, the complementary character is a T. So there's going to be a T matched with that A. For a C, we have a G complementary to it. So that's my non-coding strand that's complementary to the coding DNA sequence. A G with a C, a T with an A here, a G with a C here, an A with a T here, a T with an A, and at the bottom I have an A with a T and a T with an A. Okay. There's a directionality to these strands, and by convention we are going to write our sequences from 5' to 3' end. Again, what 5' means, what 3' means is described in the Basics of Molecular Biology lecture. The sugar backbone that is forming these nucleotides has 5 carbons, which we number 1 through 5. The 5th carbon has a phosphate attached to it. The 3' end is used to attach to the next nucleotide. So it's a long polymer of nucleotides starting from the 5' going all the way to the 3'. If you have another nucleotide, you can bind it, you can attach it to the 3'. Okay. The non-coding strand will start from the bottom. So they go in opposite directions. Okay. So for the non-coding strand, 5' is at the bottom, 3' is at the top. All right. When we write a DNA sequence down, we have to follow the convention of writing it from 5' to 3'. The sequence given to your function is written as 5' to 3'. After you identify the complementary non-coding sequence, you would have to reverse it before you write it, before you return it. Okay. So let's look at just the sequence and find the complementary sequence now. I'm going to zoom in. So I have an A, it's going to be complementary to a T, C with a G, G, C, and then A. And let me write a few of the letters from the other end. That's a T, A, T, T, A. Okay. This is coding sequence. This is the non-coding sequence. This one is written 5' to 3'. I found out I found the non-coding sequence, but right now it's written from 3' to 5'. Before I return it, I have to make sure that it's in the right order. So I'm going to rewrite it from 5' to 3'. T, A, T, T, A. So there is it. There it is. And at the end, I will have A, C, G, T. Okay. And that's the sequence that needs to be returned as the first argument from your function. Okay. So finding the non-coding sequence involves matching up A's with T's, matching up C's with G's, and vice versa. After you find that, reverse the sequence. That's going to be your first output. All right. The mRNA is where you take your coding sequence and you replace any T's with a U. Okay. The DNA has 4 nucleotide types, A, C, G, T. RNA has also 4 nucleotides, but instead of a T, we have a uracil. Okay. So the mRNA will be identical to the coding sequence, except for T's will be replaced with U's. So why is it identical to coding sequence, and how does it happen that way in the cell? When you have a DNA sequence, blue, I'll call the blue the coding sequence and red the non-coding sequence. The enzyme RNA polymerase goes in and binds the non-coding sequence. Okay. RNA polymerase is an enzyme that uses the non-coding sequence as a template, and it produces a new RNA molecule that's complementary to the DNA strand that it is using as a template. Okay. So if my coding sequence has ACG here, my non-coding sequence will have TGC. The RNA polymerase will be using TGC and producing ACG. So if you take a molecule's complementary twice, you get the same molecule. Except whenever the RNA molecule, the RNA polymerase encounters an A, let's put an A somewhere here, it will produce a U from it. Make sense? Good so far? All right. So that's how you can produce the mRNA molecule. Let's now look at the protein sequence. The protein sequence is being generated by the ribosome, which is a complex including enzymes and other RNA molecules that form a machinery that read, in a way, the messenger RNA. So it travels over the messenger RNA and using a specific genetic code translates the RNA molecule to a protein sequence. So here is what it does. Let's assume that the ribosome is sitting on this RNA molecule right here. The RNA will look at 3 nucleotides at a time. So the current nucleotide, the current triplet it's looking at is ACG. The triplets of nucleotides are also called codons. So codon is a name that we use to mean 3 nucleotides, 3 characters. After we look at ACG, we're going to shift the ribosome by 3 nucleotides to the next codon. So the next codon it will look at is going to be UGA. So it's a sliding window, except they don't overlap. All right. When the ribosome is sitting on the ACG, it will also bind a transfer RNA, which is an RNA molecule that is responsible for carrying amino acids. It will bring the amino acid to this machinery, and it will use the amino acid that is carrying. So it's carrying a particular type of amino acid. It will use that amino acid and elongate a protein that's being produced by the ribosome. The tRNA, this transfer RNA, has an anti-codon signal. This anti-codon signal will match the current codon that the ribosome is sitting on. So in this case, it will be a U, and a G, and a C, which matches ACG, right? There are other transfer RNAs that have different codons, but only the one that is complementary to the codon that the ribosome is sitting on will be able to go and bind to it. Okay. Complementarity is sort of an attachment, it's sort of a glue based on hydrophobic interactions. Only the one that's complementary is interacting, the other ones are not able to compete with it. Okay. Because this tRNA is able to go and lock itself in there, this amino acid that it's carrying will be attached to a long sequence of amino acids that's being produced by the ribosome machinery. So this will be the next amino acid. Okay. Each type of transfer RNA will carry a different type of amino acid. And the mapping between these codons and the amino acid molecules and amino acid monomers is described by the genetic code, which is shown in one of our slides. This is the structure of the transfer RNA. It has a particular region that is forming the anticodon. So that GAA will go and bind a CUU signal in the messenger RNA that is sitting in the ribosome. Okay. And this particular transfer RNA is currently carrying a phenylalanine amino acid. It's this anticodon is specific to phenylalanine. It will not have other amino acids attached to it. Only the phenylalanine amino acid is able to bind to this transfer RNA. Okay. This table shows which codon maps to which amino acid. Let's find the phenylalanine, which is right here. Okay. So a UUU in the messenger RNA or a UUC in the messenger RNA will both bind to a tRNA that is able to carry a phenylalanine amino acid. All right. So we can therefore look at the sequence of nucleotides in the messenger RNA and identify which amino acid is being produced by a triplet of nucleotides. If we see a UUU, we will know that it is mapped to a phenylalanine. If we encounter a CUC somewhere else, we will know that that should produce a leucine. Okay. So these amino acids also have single letters that are associated with them. You do not have to memorize these single letters yet. You can look at a web page or use your bioinformatics library available in MATLAB to get that mapping. Let me search Google protein one-letter code. So we have ACGT plus the uracil in our genetic code in the DNA or RNA. On the protein side, we have 20 different amino acids that are naturally occurring. They have a name, alanine. They have a conventional three-letter code, such as ALA. and they have a conventional one-letter code, in this case A. They are typically the first letter of the amino acid, but sometimes the first letter is already taken, so they use a different letter, just like the states in the United States. Okay. There are some special symbols here, the star, which means, it's not shown here in this table, some combinations of nucleotides do not encode an amino acid. Instead, they form a stop codon. They signal the stop of the translation. The ribosome machinery, as it goes over nucleotide, each nucleotide, and forms a new, combines a new amino acid to a growing chain of protein. If it encounters one of these stop codons, it stops producing and gets released from the mRNA. Okay? So the protein is produced, the production stops there. All right. So the star in our representation will represent one of these stop codons. All right. Yes.
So I know there's a stop codon, right? But then after that, there's more amino acids. But wouldn't you need a start codon for it to start coding?
You would. So in this first problem, I'm keeping it simple. I'm asking you to translate, find the triplets, and just translate into whichever amino acid it's producing. So you're not going to care about where it stops and where it starts. You will need to worry about that in the next problem where you find the gene. Okay. For this first problem, we are keeping it simple, and we are just going to find, I have an mRNA ACG. What does it produce? So we're going to look at this table. I have an ACG, right there. It's going to produce a threonine. Look at the one-letter code for threonine. It's a T. The next triplet is UGA. Look at the genetic code table. I have UGA. It's a stop codon. I'm going to replace it with a star. Next codon is AUC. AUC. Here it is. It encodes an isoleucine, whose one letter is I. So we need to programmatically go through this sequence, get the next chunk of 3 nucleotides, look at this table, find out which amino acid it is, and add it to our protein. Okay. All right. Before I look at this find gene problem, let's talk about the sort of programming skills that you need to have to solve the first problem. So you need to be able to represent a sequence. A sequence is represented as a character array, a vector of characters. So let me take this. I will call it the coding sequence equals that. Let me increase the font size. If I have a sequence, this shorter one, I need to produce a new non-coding sequence from this that's complementary. So how do I replace an A with a T and a C with a G? That can be done using if statements. There are other strategies you can use, but I'm going to describe the simplest approach. Taking the code sequence, you can write a for loop to inspect each of these characters. So a for loop starting from the first character, go to the last character, and identify what it is. With an if statement, you're asking a question. If it is an A, that means I'm going to add a T to my non-coding sequence. Okay. So let me write down for the idea for how to solve this. A rough code will go through each character in code, which means this is going to be a for loop, right? And then in your for loop, you're going to ask a question and say if the ith character in the code is an A. If it is an A, that means you need to add a T to your non-coding sequence. And then in a separate question, you can ask if it is a U, if it's a G, you can add C to your non-coding sequence. Make sense? All right. So going through means you're writing a for loop. You're going to pick out the ith character and ask if it's an A. If it is, you're going to add the letter T to your non-coding sequence. So if your non-coding sequence, if you start empty, you can add a new character to it. If you start non-empty, you can initialize it to actually be the same as code, or we will then override what the characters are. Or you can just replace the ith character of non-code with a T. So you would place T as the ith character in non-code. Again, that will be done using indexing, right? So non-code, go to the ith character and place a T in there. So this will be the overall layout for matching up one character to the other. Yes?
Is that the opposite of the template and the non-template?
Yes. So when we look at the coding sequence and the non-coding sequence, the non-coding sequence is actually the template. So this one is non-coding. It's also the template, which forms a physical template with the RNA polymerase. Okay. But this is going to be the one that's going to be identical to the messenger RNA, okay? Even though it is not physically binding the RNA polymerase. All right. After you are done with this matching process, you need to remember to reverse your non-coding variable. You can use a function like fliplr that will do reversing for you, or you can use indexing and select starting from the last one all the way to the beginning to reverse it. Or you can embed your reversing process in your for loop rather than placing your T in the ith character from the beginning. You can instead place it as the ith character from the end of non-code, which will require you to do some math to get the indexes right, but that would be another strategy. All right, once we have the non-code available, the next task is to find mRNA. Again, you would now go through each character in code. You don't have to bother checking other possibilities. I'm going to initialize mRNA to code so that it's all the same, except I will ask if the ith character is a T. If it is a T, I'm going to replace it with a U. Make sense? Because I started off with an initial state of identical to code, I'm only replacing T's. The remaining characters will be same as the coding region, the coding segment. Okay? And let me demonstrate a few of these operations in the command window. Let's say I is 5 at some point in my for loop. To extract the ith character of code, I would use indexing and get what character it is. To ask a question about it, I'm going to ask using an if statement and say if it is equal to A. If so, I'm going to go into the non-coding segment and place a T in there. Okay. In a separate question, I'm going to ask if it is a G. If so, replace it with a C. My non-code will now be, let me start off with blanks so we can see the result. Repeat this. So we placed a C with this operation. I just artificially made them all blank. This if statement asks if the ith character of code is a G. If so, go and change the non-coding I, the fifth character of non-code, to a C. All right. At each iteration of the for loop, we would be looking at one particular character of the non-code of the code variable. Using an if statement, ask a question, and if it is yes, go in and replace, set the corresponding character in your non-code variable. Okay. How do I reverse a non-code? How do I reverse a sequence? Let's look at a short sequence. How do I reverse this? One strategy is to use a function called fliplr, which will give you the reverse of text. Another strategy is to just use indexing and say, go to S, select its characters, but when selecting, start from the last one all the way to the first one. Okay? So both these strategies can be used to reverse a string. All right. And finally, let's now look at the protein sequence that is being encoded by this DNA sequence. What we're going to do now is look at the sequence. You can either look at the coding sequence or the mRNA. Regardless of which one you look at, you have to look at the sequence in chunks of 3s. So you have to set up your for loop so that your iterator jumps 3s at a time. So I will be 1 at the beginning, and then the next value of I that you want to have is 4. Starting from the first character of your mRNA, the next iteration should start from the 4th character of your mRNA, and so on. You have to pay attention to when you want to stop, right? So you have to look at your for loop, look at your sequence. Do you want it to stop at the 9th character or the 10th character if your sequence has 12 characters? Okay. If your sequence has, say, 5 characters, you don't have enough characters to make up a second codon. Your for loop will have I equals 1, but it should not try to use I equals 4. There are not enough characters left to make up a triplet. Make sense? So to make jumps of 3s, you can just set up your for loop that has 1:3: number of elements in your vector. Let's look at the mRNA, for instance. But now you have to pay attention to this upper limit, and modify it slightly to make it safe in that when you don't have enough characters, it doesn't use that location. All right. I'll leave that to you. I'll just keep my job easier and write a for loop that says 1:3: length of mRNA. Okay. You guys have to figure out how to modify this for loop so that it doesn't look at the last location if there is not enough characters left. Within the for loop, now I can use this location I to extract the next triplet of nucleotides. So if I is, let me use this sequence, this long sequence over here. If I is 7, so it can be 1, 4, at some point it may be 7. I'm gonna go to my sequence, whichever variable you are using, starting from I. I'm gonna extract that character. I will extract the next character, I+1, and I will extract the one following that. So the 7th location is right here. It has a GCT, which I was able to extract. You can extract these, all of them at once, starting from I, and I+1, and I+2. Or a shorthand for this is to say, select I to I+2. Store it in a new variable if you'd like to make it easier. Let me call this one a codon. Right. So now that I've created a new variable code that represents the current codon that I'm going to translate to a protein. I've identified the codon that the ribosome is sitting on currently. I have to now go and grab the correct tRNA molecule that has an amino acid attached to it. I have to find out which type of amino acid it has. Okay, and that can be done using a lookup, or you can use an if statement as well. A lookup or an if statement. Let's use an if statement. So you can ask related questions and say if code is equal to UUU. Okay. If so, add P is phenylalanine and P. Let's make sure. F. It's an F. Add F to my protein. All right. How do I ask this question? Is code equal to UUU? Using equal would not be the proper thing to do. When you are comparing texts that are larger than one character, you should use a function called strcmp. Okay. So again, you have a series of if questions that detect which character or which codon you have, and add the correct amino acid to your protein. And then your next question will ask, is it the next one in the table, UUC? Also add an F to protein. You can make your code more efficient and combine these two questions into one using an OR statement and say, if it's a UUU or UUC, I'm going to add an F to the end of my protein. Okay, so that's what you would do inside your for loop once you've identified the codon you are investigating at the ith location. All right. Some of the other strategies to solve this problem more efficiently and with less coding can be done using cell arrays. Okay. So if you're not comfortable with cell arrays for this assignment, just stick with if statements. If you want to practice your programming skills, try to solve the problem using cell arrays. Okay. So using cell arrays, what I can do is say, encode the entire information in a cell array. A UUU is going to be mapped with an F. On the next row, I will say a UUC is going to be encoded with an F as well. And go through each of these items in the genetic code and write down the amino acid that they are encoding. UUA is an L. UUG is an L. Now my genetic code is encoded as a cell array containing 2 columns and 20 rows. Actually, no, 64 rows, because you need to look at each combination of these nucleotides, and you get 64 different combinations. Okay, so there are 64 entries in this table. You have 3 positions. Each position can be one of 4 nucleotides. You get 64 combinations, but you have 20 amino acids, some multiple combinations mapped to the same amino acid, giving you sort of a redundancy in the genetic code. All right. Once I've set up this genetic code table, let's store it in a variable called genetic code. I can quickly, or with less coding, search within this cell array and identify for a given codon where that codon is in this table. Find by searching in the first column of this table, and then go to the corresponding second column to take out that character and add it to my protein. Okay. Make sense? Yes. MATLAB does have a dictionary. You can use that to speed up your program, in the sense that your program, when it is finding within this list where the current codon is located, can do that faster. You can use what MATLAB calls a container.map. You can look into it. So this would be another strategy if you are interested in speeding up your lookup. Okay, so you may practice that as well. It is not a requirement for the assignment, but if you would like to practice speeding things up, you can use the map container in MATLAB to perform this lookup. Okay. Another strategy to perform this lookup is to utilize MATLAB's inherent ability to do lookups in a structure variable. If I implement my genetic code as a structure array, as a single structure, whose UUU field has a value F, whose UUC field has a value F, UUA field has a value L, and UUG field has a value L. Okay. So this would be essentially a dictionary in Python terms. If I have a new codon that is, say, UUA, to identify the character, the amino acid corresponding to it, I can just go to the genetic code, which is now a structure array, and make a selection using the keys of the structure, and say select the code key. MATLAB will automatically go into that field and give me the corresponding value for that entry. Okay. So that would be another quick way of identifying the character corresponding to a codon. It requires less coding, and it's also highly efficient because MATLAB optimizes the way that fields are searched within a structure. so let's move on. That would be the strategy that you can use to do that. This genetic code is available on the course slides. You can also find this genetic code online. You can encode it in your program. You may not use any functions in the bioinformatics toolbox. If you are using Python, you may not use any functions that are not part of the basic Python language. The only function that you are allowed to use is one that encodes the genetic code, whose name is, oh, I've already defined it. Let me clear my workspace. The genetic code is actually a MATLAB function that gives you a variable containing the genetic code, which has the same layout as I described. It has field names whose fields are codons and whose values are the amino acids. Well, you have to pay attention to how these codons are given to you. They are not RNA molecules, they are DNA molecules. So you're not looking for an AAU, you're looking for an AAT. So you would need to use your DNA coding strand rather than your mRNA molecule. Okay. Rather than dealing with typing all these in your code, you can also use this function genetic code to get the mapping quickly with less effort. So that should get you going for the first part of this assignment. In the second part, we are going to look at a genetic sequence and find the longest open reading frame. So if I take a long genetic sequence and I transcribe it, this is the non-coding sequence corresponding to it, this is the mRNA, and this is the protein. This does not mean that the protein is going to be produced by the ribosome. Okay? There are additional constraints for something to be a protein. We cannot stop at multiple positions because that would sort of split up the protein into multiple chunks. Every time you have a stop codon, it's sort of, you cut through that position. So this gene would produce multiple sections, or hypothetically would produce a protein that's this long, a protein that's this long, and a protein that is that long. So the assumption is that if you have a long, very long sequence that does not have any stop codons, that is probably a protein. Okay, and this assumption is based on the evolutionary constraint that if it were not a gene and we happen to introduce a stop codon through mutations, that stop codon would not affect the functionality of this genetic sequence. Because it was not producing any proteins, just changing a codon to a stop codon should not affect the functionality of that genetic sequence as much. Okay? And as a result, you would see random stop codons for a non-coding segment, making it shorter essentially. And the longer stretches of sequences that encode proteins would sort of protect themselves from these mutations. And the protection is based on selection. If it is a coding region and we introduce a mutation that has a stop codon, that organism will not survive. It will not produce any children. We will not observe this sequence in the genetic code, in the genetic sequences, in the DNAs available currently. Okay. All right. So in this second problem, you are going to make use of the first function that you wrote to get the protein sequence, and then post-process this to identify the longest segment. An open reading frame is defined to start with an AUG. So we're going to find where AUG is located within this protein, within the mRNA. Okay. Or correspondingly, let me see if I can do this. An AUG is a methionine, and methionine does not have any other codons associated with it. So rather than looking for an AUG in my mRNA, I can instead use find M in my protein sequence. That way I don't have to deal with triplets of characters. I can just utilize a protein sequence and find out where the letter M is. So that will be a trick that I will use to simplify my problem. Rather than looking at the mRNA and finding the positions of AUGs, these are the positions. Instead of doing that, I'm going to go into my protein and find out where the methionines are. The start codon is AUG. It serves two purposes. It signals the transcription machinery to start producing a protein. It also serves as encoding a methionine amino acid. So for every protein that's being produced, their first character will be a methionine. Later, it's after the protein is being produced, it may be spliced out, it may be cut out of the protein, but to start with, every protein will have a methionine as their first character. All right. So these are my starting positions that I could possibly have a protein starting there. For my stop positions, I can either use my mRNA and ask where a UAA is. Those are the locations of UAAs, and ask where UAG is. Those are the locations of those. But these locations are not locked into the start of the codon, right? So I may have a UAA like that, starting from the second position, that is not a stop codon. Stop codon needs to be frame specific. If I'm looking at this frame, starting from there, this is my codon. I have to examine whether that codon is a stop codon. Okay. We have already handled this, this framing, this splitting into codons in the previous problem. We can again use the protein sequence rather than asking where the stop codons are. We can now ask where are the stars in the protein. That are the locations of the stop codons in the protein. All right? Okay, so now the rest is going to be a matching problem. Every start codon can be a candidate for a protein, would be a candidate for a protein, right? Let me go back and look at the indices of the methionines. My protein could start from the 39th position. if the ribosome starts from the 39th position and it continues until it finds a stop codon, where will it find the first one? 172. Okay. So the 36th stop codon, it was already left behind when the ribosome started translating at the 39th position of the 39th codon. The next stop codon it will encounter is 172. If for each start codon, I would need to look at the positions of the stop codons and find the next matching one that comes after the start codon. 39 would go all the way to 172. So one protein that I can possibly produce is going to be from 39 to 172. And this is in reference to the protein, not the DNA sequence. These are the positions within the protein. I've sort of applied a trick that way I don't have to deal with multiplying by 3 and adding 1. If I start from 116, the next stop codon is also 172. 116, if the ribosome starts translating from 116th position, it will have to stop at 172. If it starts from 198, the stop codon that comes after 198 is 246. Okay. For each start codon, you find the next stop codon and sort of record it somewhere for later inspection. Later inspection means the following. I'm going to look at this range and ask, what is the length of this protein that I'm producing? 172 minus 39 plus 1, because we're going to include the stop codon as part of the protein, even though it's not producing any amino acids for computational convenience. I'm going to have you count that star as part of the protein. Well, let's give me 172 minus 39. That is 133, right? Correct me if I'm wrong. The next segment would produce a protein that is 56 long. Okay. The next one would produce a segment that is 48 long. Okay. For each potential segment, you would find their lengths and pick the largest one. So if 133 is the largest one, that's going to be our protein that we are producing from this mRNA molecule, from this breeding frame. Okay. And then you would go back and use these indices and make a selection. Protein 39 to 172. That will be the result from your function. Okay. All right, so to make the matters worse, we are asking you to look at all 6 reading frames. A reading frame is where you start looking at the triplets. When we have a long mRNA sequence, there's no really direct way of saying this is going to be my reading frame or the next character is going to be my reading frame. The ribosome will just slide through until it finds an AUG. Okay, so we have to look at every possible location as a candidate starting position, or as a way to separate the sequence into triplets. Looking at this mRNA molecule, how many different ways can I separate it into triplets? 3. My reading frame can start from A, which will give me codons ACA, UUU, GCU, so I split into threes. That's the first possibility. The next possibility is ignoring that A and starting from the second character, in which case I'm going to obtain codons that are CAU, UUG, and CUU. Give me these codons. Okay. The next possibility is if I start from the next character A and I get AUU, UGC, and UUC, and so on. Okay. Depending on whether I start from the first character, second character, or the third character, I get different codons, and I would take these and produce proteins. So your sequence findgene function will have to generate these three different options. And then call the transcribe function to find the proteins associated with them. For each such protein, you will go through the process of finding the start codons, matching them up with the stop codons, and finding the largest one, finding the longest one. Okay? All right. To make the matters a little bit more complicated, you would need to look at the reverse direction as well. So given a DNA sequence, we do not necessarily know that that's going to be the coding strand or the non-coding strand. So we would have to look at the possibility of starting from the other end. So if this is my DNA sequence, I would have to investigate the possibility of this DNA sequence being the non-coding segment, in which case I would have to generate the coding segment from it and then repeat the process for every reading frame, translate that, find the proteins, for each one find the largest one. So I have 6 reading frames in total. Make sense? All of these 6 reading frames, you would find the winning protein that is the largest among them all and return that from your function. All right, if you do not find a gene, you would return an empty string. So if there are no start codons and no stop codons, you would return an empty string, an empty character array. If there are multiple genes with the same length, so if there are multiple winners, multiple proteins that have the same length, you can return just the first one, whichever one, arbitrarily return one of them. Okay. So that concludes my review of the programming problem.